Hello everybody, today we will talk about nerve tissue. The nervous system is essential to control functions of internal organs and systems. Also, the nervous system enables the body to respond to changes in this external environment such as reflex. Anatomically, the nervous system is divided into the central nervous system that contains brain and spinal cord, and the peripheral nervous system that contains peripheral and cranial nerves and ganglia. Each neuron has three parts. First, cell body, soma or perikaryon, three of them the same things. It contains nucleus, Nissl bodies and organelles. Soma is important for neurons, other parts for nutrition and producing neurotransmitters, and axon, the other part of neuron, usually the largest processes of the cell body, transmits impulse away from the cell body, and the last part is dendrites, are branches or short processes that receive chemical message from other neurons and transmits impulse towards the cell body. Different number of processes extend from the cell body. Different numbers of processes extend from cell body. According to it, neurons classified in three. If cell body has two processes, as you see here, if the cell body has two processes, we call it bipolar neuron. You know, B means two, so we call it bipolar neuron. Bipolar neurons have one dendrite and one axon. Sensory neurons of the retina, the olfactory epithelium in the nose, and the inner ear have bipolar neurons. The other one is multipolar neurons, have one axon, two or more dendrites. The most common neuron type is multipolar neurons in the body. And the other one is pseudo-unipolar or unipolar neurons, have a single processes that bifurcate close to the pericardium, with the larger branch extend to the peripheral ending and the other towards the central nervous system. Dorsal root ganglia and most sensory ganglia of the cranial nerves are pseudo-unipolar neurons. According to functions, neurons divide in three: sensory neuron, motor neuron, and interneuron. In this slide, you see one of a motor neuron. Motor neurons are multipolar neurons, and so you see here and multipolar neurons. This is perikaryon, soma or cell body, and as you see here from the soma there are a few processes. One of them is axon and the others are dendrites, so we call it multipolar neurons, and motor neurons are multipolar neurons. Also, you see here nucleus and nucleolus. So, motor neurons cell bodies have achromatic nucleus. Another view is from spinal cord. You see here a motor or a multipolar neuron. Soma and several processes from soma. You see soma, and from the soma, several processes are extending. One of them is axon and several dendrites, as you see here, and the soma has achromatic nucleus, so you see nucleolus. And the other one is, as you see here, multipolar neurons. How can you understand it? This is perikaryon or soma, and from the soma there are several branches, as you see here. So it is a multipolar neuron. Here you see two different multipolar neurons. This is perikaryon or soma or cell body, and from the cell body several branches are observing. And also you see here spiny dendrites. Spiny dendrites function is making synapses. Two different neurons from one of them's dendrites, others axons together occurring synapses. So axon is ending from the other one's dendrites, spiny dendrites. And also here you see two photos from cerebellum and Purkinje cells, maybe you remember from high school. And Purkinje cells are also multipolar neurons. How can we understand it? These are neuron body. From neuron body, there are several processes, several processes here. These are dendrites and these are axons. So you see here one of example for multipolar neurons. And here again a Purkinje cell here. This is cell bodies, this is dendrites, and according to section thickness you can see axons, but here this is not so thick section, so you can't observe axon, but we know that Purkinje cells are multipolar neurons. And here these Purkinje cells stained with hematoxylin and eosin. You remember hematoxylin stain for nucleus. Here is nucleus, and hematoxylin stain nucleus, and eosin stained cytoplasma. So depends on the section, as I talked before here. Depends on the section thickness, you can see more dendrites or also axon. It is 4 micron thickness, so you see a few dendrites with neuron bodies. In this two photo, we see pyramidal neurons in brain, and we know that pyramidal neurons are also multipolar neurons. If you examine it carefully, you can see multiple extensions that have come out of the neuron's body. For example, this is neuron body, and it has multiple extensions that have come out from the neuron body. Multiple extensions here, or multiple extensions are also observing here. So we can say these are multipolar neurons, and between neurons here some small points you see. These are neuroglia cells. We will talk about other slides about them. And in this hematoxin eosin stained slide, you see two neuron bodies and here glial cells neurons. Most of them are astrocytes, which is the biggest glial cell. Two of them neuron bodies and the others neuroglial cells, and most of neuroglial cells here you observe astrocytes. How can we understand they are astrocytes? For example, it is astrocyte because astrocyte nucleus is bigger than the others. For example, this is oligodendrocyte, this is astrocyte. How can I understand it? Astrocyte's nucleus is the biggest one in the glial cells, neuroglial cells. Also, it is astrocyte's nucleus has achromatic nucleus. And for example, this is oligodendrocyte nucleus. As you see here, oligodendrocyte nucleus is smaller than astrocyte nucleus. Also, it's heterochromatin. Oligodendrocyte nucleus is heterochromatin, but astrocyte nucleus pale or achromatin nucleus. So we can understand their differences by this way. Again, we can see here astrocytes and oligodendrocytes. You can compare its astrocytes nucleus is bigger than oligodendrocytes. Also, astrocytes nucleus is pale, or you can say astrocyte nucleus is achromatin. But when we look at the oligodendrocyte nucleus, it's smaller than astrocytes, and also they are heterochromatin nucleus. We see here three different nucleus type. This one, the biggest, as you see here, neuron bodies nucleus, but these and these glial cells nucleus, this one is bigger than this one. So we can bet maybe this is astrocyte nucleus and this is oligodendrocyte nucleus. How can we understand it? Astrocyte nucleus is bigger than oligodendrocyte nucleus. Also, astrocyte nucleus is achromatin, but oligodendrocyte nucleus is heterochromatin. And in this hematoxin eosin stained slide, you see abundantly smaller and heterochromatin nucleus of oligodendrocyte. For example, here especially you can understand this one and this one, this one. Abundantly we see here oligodendrocytes, and some of them, for example, this is this one, astrocyte nucleus. For example, we can look at two of them and we can compare it clearly. This is oligodendrocyte nucleus and this is astrocyte nucleus. We can understand that astrocyte nucleus are achromatin and oligodendrocyte nucleus are heterochromatin. So oligodendrocyte nucleus stain very dark, but astrocyte nucleus stain pale. Maybe you can understand by this way. So this one or this one or this one are oligodendrocyte nucleus, but this is astrocyte nucleus. Here again you see oligodendrocyte nucleus and also this is neuron body or perikaryon or soma. And here you see the smallest glia cells. What was it? It was microglia. Microglia cells has phagocytase function, if you remember, and with thin elongated and hyperchromatic nucleus. It's shaped like rod or comma. How can I understand it is microglia? Microglia's nucleus are thin, elongated, and hyperchromatic nuclei. Also, the nucleus shape like rod or comma. So we can understand it is microglia. And besides central nervous system, we will talk about peripheral nervous system. In peripheral nervous system, we call the neuron bodies as ganglion. And also, besides ganglion, there are some supporting cells, such as satellite cells or Schwann cells. Ganglion cell bodies, as you see here, oval-shaped neuron bodies, and also these ganglion cells surrounded by satellite cells. Here another view, we see three components of peripheral nerve systems. What are they? They are nerve fibers, as you see here, nerve fibers, ganglion, the oval-shaped neuron bodies, and satellite cells. They locate around the ganglion cell. When we look at the ganglion cell with bigger magnification, we see ovoid or oval-shaped cell, a chromatin nucleus, so we can see its nucleolus. And also ganglion cells are surrounded by, as you see here, satellite cells. So when we look at the ganglion, what we will see an ovoid-shaped or oval-shaped ganglion cells, and these cells has a chromatic nucleus, and so we can see its nucleolus. This is nucleolus, this is nucleus, and this is peripheral nerve cells, ganglion cells, and satellite cells locate around the ovoid-shaped ganglion cells. In a peripheral nerve, the individual nerve fibers are organized by connective tissue that consists of three distinct components called endoneurium, perineurium, and epineurium. The endoneurium includes loose connective tissue surrounding each individual nerve fiber. The endoneurium is not conspicuous in routine night microscope preparations, but special connective tissue stains permit its demonstrations. The perineurium, as you see here, the perineurium, in this schematic diagram, we can see the perineurium includes specialized connective tissue surrounding each nerve fascicle. This is nerve fascicule, contains several nerve fibers. This is nerve fascicule, this is nerve fascicule, this is nerve fascicule, contains several nerve fibers. So perineurium, this is perineurium, includes specialized connective tissue surrounding each nerve fascicule. Endoneurium is poorly vascularized, so this tissue depends on diffusion from and to the blood vessels through the perineural sheaths. And the epineurium, here, the epineurium includes dense, irregular connective tissue that surrounds a peripheral nerve and fills the spaces between nerve fascicles. So, there are three sheaths. These are epineurium, perineurium, and endoneurium. In this slide, we can observe clearly epineurium and perineurium that surround the nerve fascicles. In the slide, we observe three-layer endoneurium surround each nerve fiber. Perineurium surround each nerve fascicule. These are nerve fascicule, so perineurium surrounds each nerve fascicule. Endoneurium surround each nerve fiber. Perineurium surround each nerve fascicule, and epineurium surround peripheral nerve. Epineurium surround peripheral nerve. Perineurium surround peripheral nerve fascicule, and endoneurium surround peripheral nerve fiber. Here we see endoneurium, here endoneurium, or endoneurium, and perineurium. And in this small photo, we see epineurium. Epineurium surrounds peripheral nerve. Perineurium surrounds peripheral nerve fascicule, and endoneurium surrounds peripheral nerve fiber. Here also we see it is axon, or it is axon, or it is axon. We see axons here, and white areas locate around axons are myelin sheaths. This is axon, this is axon. This white area are myelin sheaths, and this circle are endoneurium. And here perineurium, because it surrounds the nerve fascicule, perineurium surrounds nerve fascicule, and these are epineurium, and it surrounds peripheral nerve fiber, peripheral nerves. So endoneurium surrounds peripheral nerve fiber, perineurium surrounds peripheral nerve fiber fascicule, and epineurium surrounds peripheral nerve. Thank you, listen me. Have a nice day.